glycolysis, and the role of adenine gene expression. The glycolysis pathway, there is a 6 sugar molecule, and all cells in nature use this. It is used to break down glucose, and it is essential for the metabolic process. There are a total of 10 reactions. The 10 reactions take place in 6 major steps. The first step is the addition of a phosphate group. This starts the pathway, and then after a few steps, to add a second phosphate group, the molecule is then rearranged to form the sugar fructose. The next step is the splitting of the 6 carbon molecules into 2 identical 3 carbon molecules. These are 2 identical halves of the original whole 6. The 2 halves are rearranged to form pyruvate, and while this is happening, the phosphates are removed from the adenosine triphosphate, or ATP, and this is what cells require for energy. Over the course of glycolysis, the original 6 carbon molecule produces 2 pyruvate molecules and 2 ATP. The gluconeogenesis pathway, the synthesis of new glucose, and it is essential for the metabolic process also. Essentially, it is the opposite of glycolysis. While this has a few different starting points for this process, the first step is really always the conversion of pyruvate to PEP. This is also the first major difference between the 2 processes. Overall, the systems are basically just the reverse of each other, but in this process, there are 3 major differences, the first being the one I mentioned already, and the other 2 being the removal of 2 phosphate groups, the first being from the fructose and the second being from the glucose 6-phosphate, and just leaving a glucose. The aldolase involvement involves 4 steps. It is involved in step 4 of glycolysis, and it breaks the fructose into 2 smaller molecules. There are these are dihydrohexatone and glyceride. This process takes place in 6 steps. The first step is the open ring entering the enzyme and binding with the lysine. This results in an oxygen taking hydrogen from a glutamic acid. In the next step, the arranging takes a hydrogen from and forms a double bond between lysine and carbon and kicks off the oxygen in the form of water. The third step, it forms a carbon-oxygen double bond, and in the process breaks a carbon-carbon double bond, and then it forms a new carbon-carbon double bond that breaks the carbon-nitrogen double bond. This then releases the first product, which is the glyceride 3-phosphate that leaves the enzyme. The process continues into the 4th step. The This is the lysine reforming in the nitrogen carbon double bond. This breaks the carbon-carbon double bond and takes a hydrogen from adenine, and both the fifth and sixth steps are shift-based reversals. The fifth step, water is added, breaking the nitrogen carbon double bond and adding a carbon oxygen bond, and then in the sixth step the oxygen carbon double bond is formed, breaking the nitrogen carbon bond, and the hydrogen is taken from the water, and this releases the second product, which is the dioxyetane phosphate, which is released. After this, the active site can then reform by rearranging the hydrogens and starting over. The isoforms are involved in glycolysis, fructose, and synthesis of generaldehyde and ATP. These are associated with several types of cancer. These forms are also expressed differently in different types of tissue. A is in muscle, B is in liver, and C is in the brain. This means that different ones are not only linked to different tissues, but they are also linked to different types of cancer. This in turn leads it to being used in different treatments of cancer depending on what types of cancer, depends on what type you're going to use to treat it. Thank you very much for watching this.